Our today's topic is genetics, and in that we are looking into inheritance of two genes, also called dihybrid cross. Mendel, when studied inheritance of two pairs of contrasting characters, he called this dihybrid cross. In pea plant, the dihybrid cross is this. The parents were parental generation plants were round seeded, yellow seeded was one parent. These are true breeding, and these plants produced round and yellow seeds only in subsequent generations. This is crossed with wrinkled green seeded plants, wrinkled green seeded plants. This is also true breeding. This always produced only wrinkled green seeded progeny. So these are crossed. The parents are crossed and the seeds are collected. When seeds are planted, F1 generation is raised. F1 hybrids are raised. The F1 hybrids represented by round and yellow characters only. Seeds are round, their color is yellow. Wrinkled green condition was not visible. It implies that wrinkled condition is recessive to round shape, whereas green color is recessive to yellow color. So this must be RR, this must be capital Y, this must be, because it is recessive to round shape, this must be smaller, and since the green color is recessive to yellow color, it must be a small y. The hybrid must be capital R, capital Y, small y. The hybrid must be capital Y, small y. He subjected the F1 progeny to selfing. The seeds are collected and F2 generation is raised. Since the hybrid is heterozygous for two characters, heterozygous for round shape and also heterozygous for green color, it will produce 2 to the power 2, 4 types of gametes. If you recall monohybrid experiment, where you have, this is the condition here, you have, since hybrid is F1 is RR, YY, it must produce 2 to the power 2, 4 types of gametes. If you recall monohybrid cross, where the F1 is only capital T and small t, it is heterozygous for one pair, so it must be 2 to the power 2. It will produce 2 types of gametes only and not more. Suppose if F1 is AA, BB, CC, it is heterozygous for 3 traits, so it should produce 2 to the power 3 types of gametes, that will be. So the heterozygosity is used as exponent of 2 to find out how many types of gametes it will produce. Since it is heterozygous for two pairs, it should produce 4 types of gametes, and equal number of gametes will be 4 combinations. Here is the list of gametes and their possible progeny. Here we have capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are 4 possible type of types of male gametes. Similar 4 possible types of female gametes also will be produced. Capital R, capital Y, capital R, small y, then small r, capital Y. Next we have small r, small y. F1 progeny produced 4 types of female gametes, 4 types of male gametes. When the 4 types of male gametes randomly fertilized 4 types of female gametes, this is the resultant progeny. Capital R, capital R, capital Y, capital Y. This will be round and yellow. Capital R, capital R, capital Y, small y. Due to presence of dominant genes, this also will be round and yellow. This will be capital R, small r, capital Y, capital Y. This will be capital R, small r, capital Y, small y. This also round and yellow, round and yellow. This will be capital R, capital Y, small y. This is also round and yellow. This will be capital R, small y, small y. This will be round and wrinkled, round and green. This will be round and yellow. This is capital R, capital Y, small y. This also is round and green, round and. Due to presence of a dominant gene here, it will be round and yellow. Capital R, small r, capital, here it is homozygous green. So this will be round and green. Here it is capital R, small r, capital Y. This will be round and yellow. Here this is capital R, small r, capital Y, small y. This is round and yellow, round in shape, yellow in color. This will be small r, capital Y, and this is wrinkled and yellow. This is small r, capital Y, small y. The seed shape is wrinkled, whereas the color is yellow due to presence of one dominant gene here. Here it is capital R, capital Y, small y. This is round and yellow, round in shape, yellow in color. This is capital R, small y, small y. Due to presence of dominant gene, round, it is round in shape and green in color. This is small r, capital Y, small y. This will be wrinkled in shape and yellow in color. This is capital small r, small y, and it is wrinkled and green. So 16 progeny is obtained here. And what is the phenotypic ratio in case of a monohybrid cross? The phenotypic ratio in monohybrid cross is 3:1. A dihybrid cross is the product of two monohybrid crosses. It is the product of two monohybrid crosses. So the result must be 3:1 into 3:1. It must be 9:3:3:1. This is the phenotypic ratio that is expected in a dihybrid cross where complete dominance is seen. So let us count 9. What kind of progeny will be? 9 out of 16 total. Round yellow 1, round yellow 2, round yellow 3, round yellow 4, round yellow 5, round yellow 6, round yellow 7, round yellow 8, round yellow. Out of 16 progeny, 9 are round and yellow. They represent both dominant characters of the parents. Now let us consider what is 3 ratios out of 16. It will be 1 dominant character and 1 recessive character. Now let us take round and green. Round and green, let me write it A here, round and green, B here. So this is first one, this is second one. We have round and green, the third one, ABC. So this is again 3 are there and round and green. So 3 by 16. This is verified. Now let us consider which type of progeny is 3 ratios out of 16 ratios. Here we have wrinkled and yellow. Let me write it as X. Wrinkled and yellow X. Wrinkled and yellow Y. Wrinkled and yellow Z. So this is 3 ratios out of 16. So the total progeny is 16. Out of this 16, we have so far accounted for 9 plus 3, 12, 12 plus 3, 15. The lone other different result, what it is? The last one, this let me write as, of course, without any particular symbol. So this is the last one, 1 out of 16. So 9:3:3:1 is the phenotypic ratio obtained from dihybrid cross where complete dominance is observed. It is a dihybrid ratio result where complete dominance is seen. Monohybrid phenotypic ratio is 3:1. Since dihybrid cross is a product of 2 monohybrid crosses, we can simply multiply the values 3:1 multiplied by another 3:1 to obtain 9:3:3:1. The phenotypic ratio of monohybrid cross is 3:1 and we have obtained 9:3:3:1 for dihybrid cross. And for genotypic ratio is 1:2:1 for monohybrid cross. So this is for monohybrid cross. And a dihybrid cross is product of 2 monohybrid crosses. We can, as in the earlier case, multiply one genotypic ratio of monohybrid cross with another genotypic ratio of monohybrid cross. Here, a round and wrinkled represent one monohybrid cross, yellow and green represent another monohybrid cross. The product will be 1 into 1, 1 into 2, 1 into 1, 2 into 1, 2 into 2, 2 into 1, 1 into 1, 2 into 1, 1 into 1. So this is the result obtained. Which one is represented by 1 out of 16 ratios? This is the one represented by 1 out of 16 ratios. So capital R, capital R, capital Y is represented out of 16, it is represented by 2. capital R, which is represented by two ratios, capital R, capital Y, small y is represented by 2. 2 out of 16. Let us consider other 2's also, this and also this. So this is 1 of 16, 2 of 16, other 2 of 16. This will be capital R, small r, capital Y is other 2 out of 16. And another one, capital R, small y, this is also 2 out of 16. Small r, capital Y, small y, this is the last of 2 out of 16. And we have considered which one is 1 out of 16. We have 3 such other results also. The 3 such other results are capital R, small y, this is 1 out of 16. So this one is considered for. The third such result is small r, capital Y is third such result. The last result is wrinkled and green double recessive character, that is small r, small y, this is the result. So we have seen the phenotypic ratio also. By using principle of probability also, we can arrive easily at the same fractions. Let us consider that aspect now. The same ratios can be obtained by using Mendel's probability. It was proposed by Mendel himself. And looking at the Punnett square for monohybrid cross, this is the Punnett square for monohybrid capital T, small t, capital T, small t. Its phenotype is dwarf, and this is tall, again tall and tall. To obtain the total progeny 4, and how many show dominant character? Here the dominant character is tallness, and 3 out of 4 progeny are tall. And how many of the progeny are dwarf? 1 out of 4 progeny. So recessive, this is represented by dwarf character, and this is 1/4 of the result. Now this is for monohybrid cross. Let us apply this to dihybrid cross. If we apply this to dihybrid cross, it is, let us consider what is the dominant character, round, and other color is yellow. So this is dominant character. It requires a multiplier, a fraction of 3/4. The other is also dominant character. It requires a multiplier, so it will be 9/16. This is what we obtained in the Punnett square earlier shown. Now let us consider the character round and green. Round is dominant, so 3/4, and green color is recessive, so this is 1/4. We obtain a fraction of 3/16 only. Let us consider another character. Let us go for wrinkled yellow. Wrinkled condition is recessive, so the fraction must be 1/4, and yellow color is dominant, so our fraction must be 3/4. So again we obtain a fraction of 3/16, or out of 16 progeny, 3 will be like this. Now let us consider double recessive, that is wrinkled green. This is, since it is recessive, it must be represented by 1/4. Green is also recessive, so this also must be represented by 1/4, and we obtain a fraction of 1/16. Now, so this tallies with the actual result we obtained in dihybrid cross, phenotypic ratio is 9:3:3:1. This also can be represented as the total is 9 plus 3 plus 3 plus 1 is 16. So out of 16 is 9, out of 16 is 3, out of 16 is another 3, out of 16 is 1. Now let us apply the same logic to phenotypic ratio. We have seen for phenotypic ratio, let us apply the same logic for genotypic ratio. What the monohybrid cross tells here? What the monohybrid cross tells in case of genotypic ratio? It is dominant and homozygous. TT is homozygous and it is represented by what fraction? TT is homozygous. Out of 1, 2, 3, 4, it is only 1. So a homozygous will be 1/4 in value. What about the small t dwarf? And how many dwarf progeny are there in the F2? You have only 1 out of 4. So whether it is capital T homozygous, it is 1/4, or it is small t, then also its value is 1/4. Then let us consider heterozygous tall, that is capital T, small t, and how many progeny? 1 and 2. 2 out of 4 are like this. 2 out of 4 progeny are like this. For the convenience of calculation, instead of changing this into 1/2, I retain this value 2/4. Now let us consider a particular possible genotype in our dihybrid cross of parents involving capital R, capital Y, capital Y, small r, small y. In F2, I want to know how many progeny is having, say, capital R, capital Y. And to find this out, Mendel has left us a great clue. And what is the condition of this shape of the seed? Is it homozygous or heterozygous? It is heterozygous, like our capital T, small t, it is represented by 2/4. And what about the capital Y? This is like our capital T, this is homozygous, this must be represented by 1/4. So 2/4 into 1/4 will be 2/16, this is 1/8 after reduction. Let us consider how many progeny have genotype capital R, capital Y, small y. Capital R, capital Y, small y. This is heterozygous, like our capital T, small t. So this is 2/4 into capital Y, small y is also heterozygous like capital T and small t. So this also must be represented by another 2/4. So this 4/16, and once on reduction it will be 1/4, or 25% of the progeny will be like this. Now let us consider capital R, capital Y. Capital R is homozygous, so this requires a fraction of 1/4, and capital Y is also homozygous. This requires another fraction of 1/4. So only 1 out of 16 progeny is like this. If we try to find out other such progeny, any genetic recombination, any combination of genes, let us go for small r, small y. Small r is homozygous, so it requires a multiplier of 1/4. Homozygous, it requires a multiplier of 1/4, so the value will be 1/16. You can calculate other genotypes also in a similar manner. Okay, let me take small r and capital Y, small y. This is homozygous, so it will be 1/4. This is heterozygous, so this will be 2/4. And the product will be 2 out of 16, that is 1/8 on reduction, and the total types of genotypes will be 9, that we have already seen 1:2:1 and 2:4:2 and 1:2:1. Back crosses in dihybrid cross. About back crosses in dihybrid cross, this is F1 into dominant parent, F1 into dominant parent, that is capital R small r, capital Y small y into capital R, capital R, capital Y, capital Y. This is heterozygous for 2 characters, this is homozygous for both characters. So it will produce only one type of gamete, that is one type. This will be producing 4 types of gametes it will produce. These are the 4 types of gametes: capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. And this is the lone type of gamete produced by the dominant parent, capital R, capital Y. So the first progeny will be capital R, capital R, capital Y, capital Y. The second progeny will be capital R, capital R, capital Y, small y. The third progeny will be capital R, small r, capital Y, capital Y. The fourth progeny will be capital R, small r, capital Y, small y. This is their genotype. And what is their phenotypes? This will be round, and because of this it will be yellow. It will be round, and because of one gene is dominant, it will be again yellow. It will be round, because of this it will be round, because of this it will be yellow, round and yellow. Because of this it will be round, because of this it will be yellow, again it is round and yellow. Dominant parent. All are similar to dominant parent. This is the first back cross. The F1 hybrid is crossed with dominant parent. It is heterozygous for 2 traits. So it will be producing 4 types of gametes. This is homozygous for both characters. So it will be producing only one type of gamete. All gametes having capital R and capital Y. So this is the result on random cross pollination, and this is the result. All are similar to the dominant parent. Let us consider test cross result for the same. Let us consider test cross result for the same. About the test cross result, here it is F1 crossed with a recessive parent type, and F1 is capital R, small r, capital Y, small y, crossed with small r, small r, small y, small y. It is homozygous. It will be producing only one type of gamete. It will be producing 4 types of gametes because it is heterozygous. The 4 types of gametes produced by the hybrid, F1 hybrid, are capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So this is small r, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y, small r, capital Y, small y, small r, small y, small y. What is their phenotype? We are looking at the genotype. We should observe what is the phenotype here. here the phenotype is, because of this it is round, because of this it is yellow. Because of one dominant shape gene it is round and both are recessive, so it will be green. It is because of recessive genes it is wrinkled, and because of one dominant gene it is yellow in color. Because both pairs are recessive, it will be wrinkled green. So no two progeny are similar. Of course, we have two round and two wrinkled, two yellow and two green, but no two progeny are similar. So the ratio must be 1:1:1:1. The test cross ratio for dihybrid is 1:1:1:1. This is the test cross ratio, and this is back cross ratio with a dominant parent. This is test cross ratio with a recessive parent. The ratio of parents and recombinants in dihybrid cross. Our parental combinations are genotypically capital R, capital R, capital Y, crossed with small r, small y, small y. This is parental generation, and F1 is capital R, capital Y, small y. And the F2 phenotypic ratio is 9:3:3:1. The F2 phenotypic ratio is 9:3:3:1. What the 9 ratios represent? Round yellow. 3 ratios represent round green. The second 3 ratios represent wrinkled yellow. The last one represents wrinkled green. Which of these 4 types is similar to the original parent? This is similar to original parent, and this is also similar to original parent. So parental types are 9 plus 1, 10, or simply 10 out of 16. As percentage it comes to 62.5%. As a fraction it is 10 out of 16. As a percentage it will be 62.5. And the 2 are not there in the original. They represent what is called Mendelian recombinations. And what is their fraction and what is their percentage? They represent 3 and also 3, 6. Out of what? Out of 16, 6 out of 16. This comes to 37.5. So as a fraction it will be 10 out of 16 or 5/8. This also can be written as 5/8 or 62.5. This can be written as 3/8 or 37.5. As a fraction this must be 3/8. As a fraction this must be 5/8. And as a percentage it must be 62.5. The other one is 37.5. We considered the dihybrid cross is a product of 2 monohybrid crosses. Let us look into that aspect also. Let us consider color. 2 colors are here. How many yellow progeny are there in 16? You have this 9 are yellow, not considering the other character, shape of the seed. So you have 9 progeny yellow and another 3 progeny yellow, and 12 out of how many? 12 out of 16 is nothing but 3 by 4 or 75%. So this tallies with the, then consider the other color, and that is green color. How many individuals of F2 are green in color? 4 plus 1, sorry, 3 plus 1, this comes to 3 plus 1, this comes to 4, and this is 4 by 16, this is 1 by 4. So, with regard to single trait color, it is 3:1. With regard to other trait, shape of the seed, what is the situation? Round, how many? Again you have 9 plus 3, 12 out of 16, this is 3 by 4. And how many wrinkled? This is again 3 plus 1, 4, or 4 by 16, 1 by 4. This comes to 1 by 4. So, a dihybrid cross is a product of two monohybrid crosses. The monohybrid cross for color will be 3:1. The monohybrid cross for shape will be 3:1, and their product 3:1 into 3:1 will produce the desired result 9:3:3:1. Similarly, we can verify the genotypic ratio also. It will be 1:2:1, and it also will be 1:2:1, and their product will give us. So similarly, we have 1:2:1 and 1:2:1. It will produce 1:2:1, 2:4:2, 1:2:1. These are the parental combinations. The parents are yellow and round, green and wrinkled. So round and yellow, wrinkled and green. If you look at the recombinations, the yellow gene is joining with the wrinkled condition, and similarly the green gene is joining with round shape also. Irrespective of what is parental combination, during gamete formation, if it is original parental combination or with capital R, only capital Y should join. But this is not the case. During gamete formation, you have capital R, capital Y, capital R, small y in equal proportion. Similarly, this is true for Y and also other genes also. So, basing on this, he proposed what is called law of independent assortment. Basing on this, Mendel proposed what is called law of independent assortment. When more than two alleles are involved in inheritance, the combination is not exactly the parent combination. It may be the alleles may enter any gametes, and because of this we have this is producing actually capital R small r, capital Y small y. This will be producing two types of gametes, and this is capital R capital Y, and this is capital R small y. And similarly, this is also producing two types of gametes here. This is small r capital Y, small r small y. So, if there is no independent assortment, the gametes should be having only parental combinations. So, because of independent assortment, we obtain when inheritance of more than one gene is considered, the alleles will enter gametes independent of the parental combination. This is what is called law of independent assortment. Now let us look at the problem given in one of the previous EAPMT examinations. It was given in 2004. this is the question. In a plant, red fruit, red color is dominant over yellow colored fruit. So, red is dominant over yellow, capital R and small r, and tallness is dominant over shortness, capital T, small t. If a plant with genotype capital R, capital T, small t is crossed with a plant with genotype capital R, small t, then what is the possible progeny? Here, since it is the one of the parents has capital R, all gametes will be having capital R allele or gene, and all progeny will be producing red fruit, and that is of no help in this case, because in each and every option we have red fruit, and if one of the option has yellow fruit, then we can easily conclusively eliminate that particular kind of option. Now let us consider the second character. This is heterozygous and the other plant is having both genes recessive. So this cannot be more than, so you are crossing a capital T with a small t, and it will produce 1:1 ratio. This is like, as far this character is concerned, this is like a test cross with the recessive type, and let us verify that. So the result cannot be this cannot be the result, because it is test cross result. Equal number of tall plants will be there, equal number of dwarf plants will be there. Let us verify the result. This is the gene combination. I am writing the gene combinations here. For this is the parent capital R, capital T. It will be producing two types of gametes. What are the two types of gametes? Capital R, capital T, capital R, small t. Two types. And since it is homozygous for both characters, small r, small t, it will be producing only one type of gamete, and that will be small r, small t. Now, when this pollen or egg is fertilized by this pollen or egg, we are having capital R, capital T, small t. Because it is dominant gene, it will be red. This is also dominant gene, it will be tall. Because it is, and in this combination will be capital R, small t. Because it is dominant gene, it must be, fruit must be red. And because this is recessive gene, both are recessive, this must be dwarf. So the total progeny, 50% of the progeny will be tall, 100% progeny red. Red fruit is there in all, and 50% tall, remaining 50% dwarf. So, as far this character is concerned, it is completely dominant. As far this character is concerned, it is like a test cross. So, all progeny will be producing red fruit only, and as far this tall and dwarfness are considered, it will be like a test cross result 1:1, and both types will be equal in ratio. So 50% progeny is tall, 50% progeny is dwarf, and each is having a red fruit. So by looking at the genotype, we can eliminate the first option and also the fourth option. We can easily eliminate the first and fourth options. Looking at the genotypes, capital R, capital T, small t, and also small r, small t, we can conveniently eliminate first and fourth options, and we are left with second and third. And third is the best option. That is the reason for correctness of the third option. Now let us consider the second question given in 2005 EAPMT examination. In order to find out, this is the question given in the exam, in order to find out the different types of gametes produced by a pea plant having genotype capital A, small a, capital B, small b, it should be crossed with a plant with genotype. What genotype will reveal the genotype of the crossing with which genotype will reveal the genetic constitution of the individual? Then we should go for test cross. So the answer is always test cross, and test cross means crossing with a recessive parent. So the recessive for capital A is small a, so small a and small b. So the recessive for B is small b. So the plant must be crossed with double recessive and small a, small b. This is the correct answer. So to know the genotype, genotype is revealed by test cross only. So test cross is the ideal tool to identify what type of genes are there in the organism and what type of gametes are produced by the organism. This is the best tool made available to us by great scientist, father of genetics, Mendel. In today's class, we discussed inheritance of two genes, dihybrid cross, and what is the progeny in progeny and its genotype, its phenotype, and what is the ratio obtained in test cross and also back cross with dominant parent.